Hello, I'm Commander Ian Miles. I work for the National Institutes of Health, and I'd like to discuss our results from a recent pilot study in patients with topical steroid withdrawal. It's currently available as a preprint as we await the results of peer review. So I'm sure this audience is aware of topical steroid withdrawal, but some of the features that we saw in the cohort that I'll discuss shortly is everybody had prolonged steroid exposure, typically months if not years, and then for various reasons they would discontinue, and weeks later they would manifest with this full body flushing that tended to have nasal sparing as well as the palms and soles that you could see here. And they're eczematous features, but they would have symptoms on parts of their body that they had never exposed to steroids in the first place. They describe things like red sleeves, where you see this kind of arm distribution. The term that they use for this is elephant skin, where you can see this, particularly on the joints, that the skin seems to be hanging off of them. Many would note hair loss, and then peeling, which is obviously a part of quote unquote normal eczema or routine atopic derm, but really at a level that seemed to be unusual for the disorder. Patients would manifest these symptoms. And then we could see here from an online survey that was conducted that for the most part, symptoms were fairly protracted, with only about 19% of people having resolution of these particular symptoms within 6 months of onset, and up to 25% of patients having symptoms even 3 years out after steroid discontinuation. The other unique finding here is that the patients that note resolution tend to avoid topical steroid, which is a strong argument against this being some type of underlying dermatologic disorder that they are failing to treat with the steroids. So to get at this in a more formal fashion, we took a previous publication that had given patients a fairly exhaustive survey, about 1,800 total. About 1,400 of those said that they were self-identifying as having TSW, and 400 said that they did not. We wanted to compare, were there any answers that predicted one versus the other? And so, for example, in these plots, red were the answers given by patients who self-identified as TSW, and blue are those who had eczema or some eczematous skin disease but said that they did not. It's wider up here on the top, meaning more individuals said that they had one of these higher numbers, indicating that the itch and the sleep disturbance are more severe for patients who have TSW than those that do not. But down here, in terms of the coloring on their body site, what this is showing you is that the higher numbers of red, or the higher intensity of red, means that there was more severity at the time of their initial diagnosis. And what was interesting is that those who said they had TSW tended to have slightly lower severity at the start of their disorder. I think that speaks to the fact that not everybody who has TSW started with a severe skin disease like atopic dermatitis or like eczema. Some people only had a mild rash on their hands, and that then the frequent steroid exposure compounded after a while, and then they found themselves in TSW. So some people really didn't have much skin disease before things started. We looked at all the other different answers that were provided, and what this is called is a penalized regression model, which is just a way of saying, can you use this to say what genuinely predicted who was going to tell you that they had TSW versus those who did not. And number one here is TS, meaning prescription strength topical steroids, and the number one predictor, everything that goes in this direction says it's predicting that they did have TSW. Everything in the other direction says that it's predicting that they did not. The top one saying is decreasing effectiveness. You can see the next one here is that the symptoms are spreading, needing an increased potency, which is another way of decreased effectiveness. Those are the top three. So those all speak to the idea that you're using steroids, and even though you're using them, it's not working. The effectiveness is wearing off. The rash seems to be spreading. You need higher and higher potency to try to get some kind of control. That was the top kind of signal that things were problematic. The first symptom that was on its own was that temperature dysregulation, where you could feel too hot or too cold, followed by redness and burning. You also had even over-the-counter strength, so hydrocortisone here, the same thing. Like, do you have to have spread during hydrocortisone use? That was problematic. Protopic is on this list, and we're going to have to do follow-up studies to figure out what that means. Does that mean that people who had TSW had a severity of skin disease that eventually landed them on protopic, as in somebody saw their skin disease and thought, all right, well, steroids aren't working, why don't we try a calcineurin inhibitor? Or is there some potential that, calcineurin inhibitors worsen your risk of TSW? We don't have an answer for that right now. One of the things that was predicting the opposite direction, so, would you not have TSW, is if you had hydrocortisone exposure, but no symptom worsening. This kind of makes sense. So if over-the-counter strength steroids were controlling your disease, you probably don't have that protracted exposure to topical steroids or ramping up the dosing or ramping up the potency. Everything's fine at the over-the-counter. You probably use it for a little bit and move on. Dryness actually predicted the opposite. So that would indicate that people who have TSW might have skin that isn't as dry as you would expect for atopic derm. So like I said, number one, escalating steroid use spread despite using steroids was the most predictive factor. So we can then take all of those and try to put them into what would be called major or minor criteria. You do this to try to come up with a way that you can just kind of evaluate each patient individually and say, all right, what are the chances that they really do have TSW? So the top predictive ones were the burning and flushing and temperature dysregulation. And we can separate them out into less common or less skin-specific symptoms. And this matrix here is just basically saying, if you took everybody in the trial, and let's say we said, all right, you had to have literally every single possible symptom of TSW. So all 3 major and all 9 minor, that's 25% of people. So 25% of people who said they had TSW have every single one of the symptoms. But you're trying to capture at least 90%. That's what the goal here is. So if you look at this one here at 92, that would say if you have one of the 3 major and 3 of the 9 minor, then if you said, all right, that's what we need to give somebody a diagnosis of TSW, then you would capture 92% of the people in that survey. Now, this of course would mean in theory you would be failing to diagnose 8% of the patients with TSW, but this is kind of how these systems are created. So if you looked at this and you said, hey, look, I have flushing, I also have bone deep itch, loose skin and these zingers, that would be sufficient to diagnose with TSW. So we brought these patients in having this understanding of what the clinical picture should look like, and then we ran them through multiple different analyses using a host of collaborators I'll mention again at the end from across NIH. And the first evaluation is the microbiome, which is what our lab primarily works on. What you're looking at here is each column is an individual. So these are the healthy volunteers, and then these are patients with TSW for comparison. Every color is a different organism, so this would just mean that this individual has a whole lot of this very dark purple, but all of the healthy volunteer people have very little of that particular organism. So the ones that are dominating across this is really Staph aureus is the most stark one. So here in TSW is blue, so they have a higher amount of Staph aureus than the healthy volunteer, and they have a lower amount of what we call just biodiversity. So really, if you look at it, you say, all right, they have too much Staph aureus on the skin, and that's very much like atopic dermatitis. I very much do not think the staph is causing their problem. There's quite the controversy about just how much staph is, a freeloader on inflamed skin versus something that's genuinely causing the problem. But nonetheless, if you had shown me their microbiome picture and said, what disease do these patients have, I would have guessed, quote unquote, normal atopic derm. The other thing we did is look in the serum for various inflammatory cytokines. I'll tell you, these are not the kinds of tests that you would be able to get at a regular doctor's office. These are special assays that you have to have a particular piece of equipment for. But you can see here's healthy volunteer and TSW. So the higher the number, the more of that cytokine you have. And so this is IL-13. This is one of the two cytokines, IL-4 being the other one that dupilumab blocks. So IL-13 is up in these patients. It's also up in the comparison group that we use is patients with atopic derm who did not have any reason to think they had TSW. IL-6 is another one. IL-4 was up. So basically, these are just inflammatory cytokines that are upregulated. And once again, if you had shown me their inflammatory cytokine picture, I would have guessed, wow, these people look very much like atopic derm. The next is we wanted to see what kinds of cells infiltrate the skin. So when somebody says, oh, your skin is inflamed, what they mean is that you have a lot of immune cells in the skin. And so these are just one kind of immune cell. This is a representative image for one of the patients with TSW. These are CD4 T cells. And so you see a lot of them just with one cut through the skin. That video you saw there on the bottom is a healthy volunteer. We're staining for multiple different ones. If you just take a slice from a healthy volunteer, it would look like a black box in the sense that you wouldn't see any green because there's so few T cells in a healthy volunteer skin that just doing one slice across, you're not going to be able to see it. The only way you can see the cells is by making these kinds of movies and stacking them up. So this tells you that T cells and macrophages, these are the kinds of cells that are in the skin of patients with TSW. And once again, I would say, wow, that's very much like atopic dermatitis. So the take home from this is that they have allergic inflammation. That allergic inflammation does not look any different than any other form of allergic inflammation that you might see. But that is where the abnormalities stopped. We also decided to look by metabolomics and transcriptomics. So metabolomics is you're trying to look through all the different metabolites in the body at the same time. So let's say you ate an apple and we wanted to look down the line to say, all right, where are the sugars, where are the fibers, that kind of stuff. Those are the metabolites. And transcriptomics just means that you have genes that have to be turned into proteins. So you go from DNA to RNA to proteins, and then those proteins have effects downstream. So the RNA part is what you're trying to look for. You're saying what genes are actively being expressed in the body at any given moment. And so the way that these graphs work is down here in this corner, this would mean that there'd be no difference or zero. And as you go up towards this direction, this would make it more significantly different between the two groups that you were looking at, and in various different ways. More metabolites are different, and then the higher up in the graph means that they're further apart, or there's a bigger, more important difference between them. And so if I take healthy volunteers and compare them to quote-unquote regular eczema, like people who have eczema but don't have any reason to think they have topical steroid withdrawal, that's what's in the not bold. So nucleotide sugar metabolism, urea sugar, and various lipids will show up every time somebody looks between those two groups. But if I take TSW and compare them to either healthy volunteers or atopic derm or both at the same time, this thing gives up vitamin B3, there's nicotinate and nicotinamide, and vitamin E abnormalities. So you have too much of that in the skin of those patients. So this is different. So vitamin B3 is not something that has ever shown up in a evaluation of what metabolites are abnormal for regular eczema. The things that are down, the same thing. The things in bold are unique to TSW, and you can see that the most significant down regulation is in tryptophan metabolism. And so you want to look at all right, well, what would connect those two? And this is just one example of how those two pathways intersect. So as it would turn out, tryptophan here, TRY is the abbreviation. So tryptophan is made into nicotinate, nicotinamide, and in this one, NAD plus is the abbreviation. The first step in that process involves these two enzymes. This one, TDO, is a well-established target of glucocorticoids. Now, glucocorticoids are the more scientifically accurate term for the types of steroids that you would get in a quote-unquote topical steroids. Right? So you might have heard the term topical corticosteroids, topical steroids, but glucocorticoids are the more official molecular term. So this just means that your topical steroids are known to induce the first step of turning tryptophan into niacin or nicotinic acid. Along the way, you generate these things in the middle called kynurenins, Now, this particular publication is about the brain inflammation, but there's many others that have shown that kynurenins are neurotoxic and they're anticholinergic. So this A7N-AChR, that is one of your cholinergic receptors. The cholinergic system, you might have heard, it's called parasympathetic, so it's the opposite of sympathetic. So if you've heard fight or flight, that's the sympathetic nervous system. Rest and relax is the cholinergic and kind of parasympathetic nervous system. So if you're blocking the choline, you're blocking the ability of the body to kind of rest and relax. And so the metabolite that the patients had too little of is made into the metabolite that they seem to have too much of. And niacin, nicotinic acid, used to be used a while back for cholesterol metabolism, but they stopped because it caused so many symptoms in the skin. They used to take niacin and people would have this massive flushing and they would have skin disorders. And so it's really fallen out of favor. So this jumped out at us initially as that kind of makes sense, at least for their flushing disease. And where, NAD and niacin comes from in your skin, we got to go way back to biochemistry and talk about the electron transport chain. I think most have heard, the mitochondria is the powerhouse of the cell. And in order to make the powerhouse to generate our energy, it's actually turning electrical energy into chemical energy. And so here is a video from Bioman Biology. You can find the full video on YouTube. So NADH is the molecule that eventually is carrying the electrons to part of what's called the electron transport chain. And then these E's just slide over from complex 1 to 2 to 3 to 4 and will eventually turn oxygen into water. So this is why you need to breathe. The oxygen comes in and it's pumping hydrogen or acid into kind of the middle of the mitochondria, and then the acid flows back in, and genuinely almost like a water wheel, the physical turn of the ATP synthase helps us make energy. So electricity pumps acid into the outer part of the mitochondria. The acid flows back through a water wheel, and that is how we get our energy. Kind of wild, but that's where NAD comes from. And so then when we look at transcriptomics, again, this is kind of asking us, all right, what is the body actively making? This is the more scientifically fancy way of drawing those same kind of blobs in the electron transport chain where the electricity and the hydrogen were flowing through. So this is complex 1. This is that first one where NADH turns into NAD plus. And in everything you see in red means that there was more of it in patients with TSW. So all of these here are the different components, and this one as well, of complex 1. So they have way too much of complex 1 compared to patients with either healthy volunteer or patients with atopic derm. This graph over here is that breakdown that I showed you in the one that talking about the brain. So this is how you get tryptophan into niacin, and here's those first ones. This is the TDO that is induced by steroids, so the enzymes that are breaking it down. So what we perceive here is that the tryptophan in the body has to be turned into NADH. That is then turned into nicotinic acid by complex 1 of the mitochondria. And I'll show a summary of this at the end. So they have too much niacin, too little tryptophan because they're breaking it down to generate this and churning it through the mitochondria and the electron transport chain. There's absolutely nothing like atopic dermatitis. There's no shortage of transcriptomics or metabolomics studies in atopic derm. If you look at atopic derm, it's almost entirely about ceramides and sphingolipids. That's where those abnormalities lie. They do not lie within the mitochondria. We can check this in another way by another collaborator, Louis Franco at the National Institutes of Arthritis, Musculoskeletal, and Skin Diseases at NIH. And what they did is they brought people in and they put steroids on them, healthy volunteers. So you do a biopsy, then you put steroids on and you wait 4 hours, just 4 hours, and then come and do another biopsy. And what you're seeing here is the cartoon drawing of the mitochondria, and these 4 blocks right here are those complexes, those little blobs that were accepting the electrons and letting the the protons, the hydrogens go through. And so you see here CX1, so this is complex 1. So within 4 hours in healthy people, you already see an upregulation of complex 1 of the mitochondria, which as a consequence would be expected to start churning out an increased amount of niacin or NAD+. So, I said before that niacin is a pretty well-known inducer of skin flushing, but those studies were all done about people taking it as a dietary supplement or as a medication orally. Not really a whole lot had been done about, what does it do on the skin or in the skin. And so what we're looking at here is we put the niacin directly onto an ear of a mouse, and we're trying to measure how much swelling there is. So the higher the level here, that means there's more swelling that's induced. So, higher doses of niacin directly onto the mouse ear will induce swelling. But the part that was a little odd at first when we saw this, so this is just what a mouse ear would look like if you didn't do anything to it, except right here, I've kind of shaved. You almost can see the grooves from the shaver here. So I've shaved down so you can kind of see beneath the line of where this is normally covered by hair in a mouse. And this is where I added the niacin, and the redness you could see is really restricted to the part below the hairline. So if I just left this with the hair, it'd be very difficult to see anything wrong with the mouse ear at all. And you compare this to what I would refer to as a more standard model for atopic dermatitis. So if you want to give a mouse eczema and then take away the eczema using drugs as a part of an evaluation you might go to the FDA with, this would be the model you would use. And you kind of get the sense that this dryness and thickness and abnormality is all over the ear and not just below the hairline for these mice. And you now combine that with the fact that we discussed at the very beginning, the patients don't often have any symptoms on their nose, on the palms of their hands, or at the bottom of their feet. Those are the only parts of your body that are completely free of hair follicles. And hair follicles are intriguing in the skin because that's where your stem cells reside. So this is kind of a cross-section of a hair follicle, and down here at the bottom is where all the stem cells live, and they make new cells. So as these cells differentiate, they will crawl up the hair follicle and then move out, and that's kind of how your skin replenishes itself, which is intriguing because if you sit here today, every single cell in your skin is different from what it was 45 to 60 days ago on average. So that means every cell in your skin, those keratinocytes, fibroblasts, all those different kinds of cells, has died and been replaced by one of these stem cells. The only cell type that hasn't changed from only skin cell type are those stem cells. So it was intriguing because if TSW was just an abnormality of keratinocytes or fibroblasts, or kind of the normal cells, it shouldn't last for more than 2 months. Every cell should be reset. You stop using steroids or whatever the offending agent might have been, and everything should be reset in 2 months. So the fact that it would take some people years stood out as something abnormal had to be occurring. So we thought, well, maybe if you are corrupting the stem cells, what that's doing is it's destroying the original copy. And so then every copy you want to make after that will have that abnormality with it. And, hopefully, over time, your body might be able to repair things and put it back from whence it came, but that might be why it takes so long to go away. So if you take these stem cells, you can just buy them from different companies that just sell them kind of perpetuated to this point. And at higher doses, it's kind of hard to see at this magnification, but you get the sense that there's a denser picture. And so what you're doing is you're throwing the nicotinic acid in with these stem cells, and you're seeing it's causing this upregulation and this proliferation. And then we can measure to see all the different kinds of cells you might find in your skin that would be relevant. This means human follicular stem cells, so that's these guys down here at the bottom. KC is keratinocytes. That's what you normally think of as a skin cell. Like if I started just rubbing your skin and things start flaking off, those are corneocytes that came from keratinocytes. Fibroblasts are just below that, and then Schwann cells are a type of nerve cell that is prevalent in the skin. And the color coding here is just the more orange and red it is, that means that particular metabolic pathway was more disrupted. And into the cultures, what we threw was either clobetasol for 24 hours clobetasol for 48 hours. That's what steroids, that's what the S is steroids, or clobetasol for 24 hours and then take away the steroids for 24 hours. So thus kind of a cellular withdrawal, so to speak. Now this is only 24 hours. Obviously the patients are more along the lines of months, but you can see that there's a difference between how cells behave between having continuous steroid exposure versus having steroids and then having them removed. So these stem cells, the induction of that vitamin B3 pathway is much stronger if you take the steroids away than if you had left the steroids in there across the board. So putting this all together, here's what we think is going on. So the corticosteroids, the glucocorticoids, officially, this is a cartoon of the mitochondria, and normally there should really only be one of each of these as a set, but what happens is, for whatever reasons, the complex 1 duplicates itself. So now the patients have way more complex 1 than they're supposed to. What complex 1 is supposed to do is take NADH and turn it into NAD+. And NAD+ is, again, related to niacin. That would explain the flushing, and anhidrosis is a fancy word for saying that they have difficulty sweating. Sweating and things that make them hot might make it worse, but during the peak of the symptoms, patients typically report that their skin didn't sweat the same way that it once did. So niacin would create that flushing, initial flushing, and the loss of sweating. It induces a pathway that, this is Wnt beta-catenin. Without getting into too much detail of this, there are no human beings in existence that have this pathway dysfunctional, because if this pathway doesn't work, you don't make it out of the embryo stage. But this pathway is necessary to activate to make new skin. And so if you overactivated it, you would anticipate a bunch of cell proliferation. So those cells would just start growing uncontrollably. Wnt beta-catenin, as you might guess from what I just described, is often upregulated in cancers, in terms of those cancers being overactivated. This is not activated to that degree, so we're not referring to this as a cancer type level, but Wnt and beta-catenin would cause hyperproliferation of the cells. And to us, that might explain those elephant wrinkles where the skin is just growing beyond its borders, and so it's kind of sloughing off and hanging off of the joints. And it would also explain just this profuse amount of cell proliferation and the shedding, where patients are telling us that they come home from work, they take, one said that she takes her bra off and like a cup of cells fall out just because this massive amount of skin shedding coming from that could only be explained by pathways such as this. Now, in order to make the NADH to go through complex 1, you got to break down tryptophan. And again, to do that, you break it down into kynurin, which is neurotoxic to various degrees and would explain the zingers and those other kinds of, the better term would be, a more official term would be neuropathic pain. Kynurin blocks choline, anticholinergic, and thus would also contribute to that flushing and lack of sweating. Kynurin induces a receptor called TRPA1, which our lab works on quite a bit. This receptor's official job is to kind of help you tell the temperature. It's to report back to the skin what the temperature outside is and then adjust accordingly. It also relays signals of pain and itch, so it will pick up irritants. So this would make patients more susceptible to the kinds of pollutants that I've discussed on this channel before that will activate that particular receptor out in the environment, so they might be more sensitive to the external world, but it also explains some of the skin pain, that really deep itching sensation, and the sensation of thermodysregulation, because one of the key receptors needed to properly assess the temperature is now basically disordered. The other thing that happens when complex 1 is overactive is you generate reactive oxygen species. So if you've heard the term antioxidant, you're taking an antioxidant to stop ROSs, or reactive oxygen species, from doing harm. If you don't have those, then these particular inflammatory pathways are upregulated. Now, MAPK and FKB, these are not unique to TSW. This is what I was saying in the very, before, saying that it's just they have inflammation. They have the kinds of inflammation one would anticipate having for this kind of allergic skin disease. What is abnormal is this niacin and this neurologic focus of their disorder through kynurin and niacin. So what you want to do, or what we thought to ourselves is, all right, well, if the root of the problem is too much complex 1, what can we do to block that? And there was at least one drug that's world famous for its ability to block complex 1 that people would use it. People would use it in laboratory settings just to prove to people, hey, look, I block complex 1. There are multiple ones you could pick from that would be better termed lab chemicals, but the only one that's a real drug is metformin, the anti-diabetes medication. And then we started poking around too and found, berberine is sold to some extent as herbal metformin, and so either one of those might have that effect. And when we look back to the literature, there's really only 2 clinical trials where anybody took patients with TSW. I will say they didn't rigidly define who classified for TSW. They didn't go through and say, like, all right, how many major, minor criteria, but nonetheless had people who, for the most part, seem like they have TSW. One of the trials was dupilumab, we'll talk about at the end, but the other one is a traditional Chinese medicine whose primary ingredient was berberine. So it was a successful trial. Berberine was one of the second most common ingredient in this complex mixture, also well established as its ability to block the mitochondria. Now, berberine has gotten quite a lot of fanfare, most recently because of TikTok, social media level claims referring to it as nature's Ozempic. I don't know. The data claiming that it would have Ozempic-like effects is not persuasive at this stage, but very persuasive that it will block complex 1 of the mitochondria. And you can actually watch that happen here. So OCR stands for oxygen consumption rate. It is a instrument that you would measure the mitochondrial activity. Now, you're not separating the different complexes, but you're just saying how active is the mitochondria. And so here in black, or the squares, is berberine. And so at higher doses, across all the doses, berberine is better at shutting off the mitochondria than metformin is, but both of them do have the ability to do so. Down here at the bottom is ECAR, extracellular acidification rate, which is a marker of glycolysis, so breaking down glucose, and metformin is far better at that than is berberine. Whether that has any statement as to how much better metformin is at controlling blood glucose, I don't know, but we're intrigued by the fact that, all right, berberine can inhibit the mitochondrial function. In the cohort, some chose to go with metformin from their primary care provider, while others chose to have berberine either over the counter or over the internet. The dosage that was used is people tried 1 tablet or 500 milligrams a day for a week to make sure that their body responded well. As you can imagine, this is a patient population very concerned for the potential for drug complications. After a week, they would escalate up to 500 milligrams twice a day. There were a few in the cohort that went higher than that and did not report any additional benefit at those higher doses. So I don't really know if higher doses would be of any greater utility. But we can say that metformin had about just under a 50% improvement on average, whereas berberine was upwards of 70-75%. Those differences are non-significant between the two. One intriguing finding was the individual here in the open diamond started on metformin and then changed to berberine, noting a much better improvement there. The individual here in the triangle used both berberine and a light-based therapy that's subtly different from phototherapy. So both were started at the same time, so we can't officially say. And then the individual here in the open circle only was on treatment for about 3 weeks, discovered she was pregnant, and then out of an abundance of caution, stopped. So you can see the before and after. People were quite happy with mostly their improvement, and you can calculate based on that survival plot of symptoms versus the people in our cohort what we would have expected in terms of just natural resolution if we had just followed people for 3 to 5 months, which was how long these interventions kind of went on for, versus what we observed. So a significant improvement, no doubt. So a quick overview of everything. We think we're using our clinical evaluations to come away with the notion that TSW seems to be marked by this protracted, often neuroinflammatory symptoms, most often marked by escalating steroid use and requirements. Using metabolomics and transcriptomics, we could say it's associated with mitochondrial complex 1, oxidation of NAD. Mouse and cell models teach us that NAD induces a glucocorticoid responsive inflammation, and that glucocorticoids can induce NAD, so there's a reciprocal relationship there. Functional assays tell us that glucocorticoid inflammation can be modulated by complex 1 inhibition, and so we take this information back into the clinic, and that would inform a case series on complex 1 blockade, which appears to at least have some clinical activity. So there are some obvious limitations. Our diagnostic criteria, we do not have specificity. So we don't have a large number of patients with self-identified atopic derm who deny having topical steroid withdrawal to say, well, how many of those people would have met that criteria. So we will need to derive something of that. Obviously, it's pilot. It's open label in the case series. It's a small N value. And so that always comes with those particular caveats. There is the severity, as I said, just overall skin disease was much higher in our cohort than in atopic derm. But I will note that the literature is not scant on the metabolomic, transcriptomic, cell infiltrate, microbiome signals of atopic derm. So we can at least compare what we are finding for TSW for the established literature. So what I would say are knowns is that really it's eczematous inflammation, but it is not to be confused with eczema, an overproduction of vitamin B3, niacin, nicotinic acid through complex 1. The overproduction of neurotoxic intermediaries is driven by tryptophan breakdown. So because of the role, the upregulation of IL-13 and IL-4, there may be a role for dupilumab, and there's at least one controlled trial looking at dupilumab as efficacious therapy for TSW. Berberine, and to a lesser extent metformin, the improvement there was less impressive, but maybe deserving of a larger trial. Things that we don't know, we looked quite extensively for the types of biomarkers that could be run in a normal clinic. It's one thing to find a biomarker in the serum using mass spec, but you really need to find something that people could send off to a local lab, and we have yet to identify that easily. The correct dose, there's really, on the internet, there's doses that range into, quite absurd. And anytime you're dealing with something over the counter, you have to be cautious about whether or not what they're claiming is actually in the bottle. So before we had discussed any of this with the patients, we looked up on the website Consumer Lab, and you could see only 2 brands had the level of berberine that they claimed to have on the bottle, Natural Factors and this one called Solar Ray. We don't endorse any particular one, but just note that you got to be cautious. Just because your patient is buying something that has berberine on the label does not mean that they are taking that at all. Other questions would be, should we be using this topically or orally? Oral seems to have some effect. It is incredibly yellow as an herb, so if you were to just throw something into a bathtub, for example, that would be pretty discoloring and would be a much higher dose than it seemed that the cell cultures needed. So if one, for example, were to follow the model of bleach baths and take 1 capsule and dilute it in a liter of water, and then from that initial batch take 1/4 to 1/2 a cup into a bathtub, you would end up with the concentration that worked fine for a cell culture. Whether that would work topically is speculation at this time, but something that might be worthy of a targeted study. How long would somebody have to take it if they feel better after a few months? Is it okay to come off of any of these blockades? There are other speculations one could do if you were attempting to target complex 1 of the mitochondria. just pure heat. This is distinct from phototherapy, which may also generate temperature, but just actual heat will inhibit complex 1, so that's something that might be considered. Isotretinoin was mentioned on a recent video from Dr. Lazewski as well as Dr. Leo. Dr. Leo was the one who ran the dupilumab trial for TSW, and Dr. Lazewski in their conversation had mentioned isotretinoin as something that he uses. That, by the way, does inhibit complex 1 of the mitochondria, at least in cells. And really the most stark known unknowns are, why does this happen to some people and not others? Clearly, the majority of patients will use topical steroids, sometimes even chronically, and not have anything close to the disorders that these patients had. We do not have a great answer for that. And can this be prevented? And as Dr. Leo articulates very nicely in this video, which I would recommend if you're interested in this topic, you also check out theirs. Really it's stewardship of steroids and making sure that people are not on these topical products for 4, 5, 6 months or more in a row, and being cautious about when you have a patient who you've noticed continuing escalation of topical steroid needs. It might be time to back away and try a different agent altogether. So once again, all of this information and much more is contained within our preprint, which will hopefully be a peer-reviewed paper at some point, although that process tends to take a while. For those who are not familiar with these sites, if you need help finding it, there's that QR code right there. But this really represents a lot of different laboratories collaborating across the National Institutes of Allergy and Infectious Diseases at NIH, as well as NIAMS, and some collaborators from Allergy and Asthma Network, as well as ITSAN. So I want to thank everybody for their attention.